we try to help with unbiased analysis of different therapeutics, and we advocate smart trial design, like using a core protocol and sharing data to address recruitment challenges, and we do that in close collaboration with patient advocates. So the first subject I would like to discuss is a consensus-based expert recommendation for the diagnosis and clinical management of vanishing white matter. And some of it was addressed this morning, that it's very difficult for people in when they go to like the doctor and emergency rooms to get across what's needed for vanishing white matter. So we decided as a consortium to work on that and come up with recommendations, and the paper, I think, looks like it's going to be published pretty soon. So we did that. Participants in this effort are the Vanishing White Matter Consortium. We also involved other experts, like other leukodystrophy experts, specialists from relevant medical fields, including geneticists, intensivists, dietitians, endocrinologists, a pediatric gynecologist, and a complex care pediatrician. And we involved patient advocates, like the Vanishing White Matter Families Foundation, the United Leukodystrophy Foundation, and the VKS in the Netherlands. We used a multi-step approach, and we consulted experts and patient advocates to identify the topics to be discussed in the eDelphi procedure and to formulate draft recommendations. We did a systematic literature review to collect all available evidence substantiating or opposing the draft recommendations, and PhD students did that. We executed the real-time eDelphi procedure to agree on the recommendations. So we worked with an extended expert panel, as explained above, for that subject. And we agreed on lay language material, including a patient management card, and that we did with the Vanishing White Matter Consortium and patient advocates. And everybody was involved in the final draft of the paper. So we made recommendations on how to diagnose patients based on MRI criteria and on genetic criteria. You don't see clinical criteria here. The diagnosis is based on MRI and genetic criteria. We said something about the prediction of the disease scores, and what's clear is that the age of onset is the critical factor predicting the disease scores. We made an overview of measures to prevent episodes of rapid decline, like trying to avoid fever and infections, apply vaccinations, avoid head trauma. And something that might be important is standard behavioral management. So the stressors that need to be invited do not include psychological stress. Standard behavioral management of children is advisable. Avoid ISR-provoking medication, and we made tables of that. And what's important, that the only clinical and laboratory evidence for concern the risk of inhalation anesthetics, especially sevoflurane, and with propofol as a safe alternative. That will be in writing, because if a patient, all the time I press the wrong button. If a family says to a doctor, you cannot use sevoflurane, but please use propofol, then the doctor will most likely, the anesthesiologist will say that's for me to decide. But we put that now in writing, and actually we also published a paper on the subject, on a separate subject, so on anesthesia. We made an acute presentation management plan, and also a chronic disease management plan. So hopefully the paper will be published soon, and then it will be published also on the website of the Vanishing White Matter Consortium, and hopefully also on the website of the United Leukodystrophy Foundation, so that it's accessible for everybody, and you can present it to your doctor, so that it's I'm not sure what I'm doing. So this is the management card as it is on two sides as we drafted it. So it says something about the disease, that has for the doctor do's and recommendations for the patients and recommendations for the physicians. and also how to contact us, and where to find more information, and who established this leaflet, and so that it's clear that it's official. You cannot put it yet on any website, Allison, but hopefully it's published soon. Hopefully in a couple of weeks, actually, because it has already been reviewed. So I think that this might help very much with daily life for families to get around with their doctors. Something we discussed this morning, and I think that should be done for many more disorders. Then we made Vanishing by Matter conditional mutant mice, and I would love to share this with you because it comes with implications for how to look at MRIs. And this is a slide on what pathology in Vanishing by Matter patients looks like, and you've heard it before. I just summarize it. So astrocytes and oligodendrocytes bear the brunt of the disease. They look abnormal. They are immature. They fail in their function. And so oligodendrocytes make myelin. There's lack of myelin. Astrocytes make sure that volume regulation and iron and water homeostasis is maintained. And if that's not happening properly, then myelin gets vacuolized, and there's no scar tissue, and in the cerebellum, Burke glia are mislocalized. We made mutant mouse models, as you know, and they completely recapitulate the pathology and also the clinical disease. So what you see here is normal myelin stain. Then this is the milder mouse. You can see all the vacuoles in the white matter, and this is a severely affected mouse, and you can see how much vacuolated the white matter is. We actually wanted to know what the contribution of each cell was to the pathology of vanishing white matter, so we made cell-specific mutants, and the first two are astrocyte-specific, so they have vanishing white matter only in astrocytes. Then this mouse has vanishing white matter defect only in oligodendrocytes, and this mouse has it only in neurons. If you look at the myelin pathology, then this is normal myelin. Then you can see here the whole body mutant. So this is the mutant vanishing white matter mouse with lots of vacuoles. And if you then look at the cell-specific mutants, you can see the vacuoles in the astrocyte-specific mutants, but not in the oligodendrocyte-specific mutant and not in the neuron-specific mutants. So actually, this is something you understand, because astrocytes are working on iron and water homeostasis, so they are responsible if they are sick for the myelin vacuolization. Then we looked at myelin pathology, and specifically, and what you see is oligodendrocytes fail to myelinate all exons. If you look at the EM of the astrocyte-specific mouse, you can see that the myelin looks awful. Like it looks like it contains vacuoles and water within the myelin sheath. If you look at the oligodendrocyte-specific mouse, if the myelin is there, it looks beautiful. No vacuoles in it, which is in line with that the astrocytes are not sick in this case. But what you do see is that many exons don't have myelin at all. So there's a problem with the amount of myelin in the oligodendrocyte-specific mouse. So in astrocyte-specific mouse, we have vacuoles in the myelin, and in the oligodendrocyte-specific mouse, we have lack of myelin. Oligodendrocytes, I'm not good at this. Okay, oligodendrocyte pathology. I think this is hypersensitive to my thumb or something. I didn't even touch it. Oligodendrocyte pathology, the mutant oligodendrocytes fail to mature. That's something that we expect. And astrocyte pathology, if mutant astrocytes are immature and don't perform their function, mutant Burke glia are mislocalized, and mutant oligodendrocytes induce astrocyte reactivity. Now then the question is, what do these mice clinically look like? And I'm not sure how to activate the movies. Can you activate the first movie? That's the wild type? Yes. Beautiful. So that's a normal mouse. Walking on the catwalk. Can you activate the second one, which is the whole body mutant? So that's the full mutant mouse. That's very ataxic. You can see it walks much more slowly. It's leaning against the wall because of instability. And can you then activate the next one? Yes. The oligodendrocyte-specific mouse is equally miserable, very ataxic. And can you activate the astrocyte-specific mutant? Which is much better. So, to my surprise, because we thought that the astrocytes were most important in the disease, but actually it's the oligodendrocytes. And you can just reflect it here. So this is the ataxia score, which is in the whole body mutant are the gray dots. You can see the severe ataxia, and this is the oligodendrocyte mutant, which is equally ataxic. The rest, the astrocyte mice are these two mice, and you can see one is completely overlapping with the control, and the other one is a little bit elevated. So that means that mutant neurons do not significantly contribute to the clinical phenotype. This is something that we expected. Mutant astrocytes induce all sorts of problems, including myelin vacuolization, but neither is associated with a significant clinical phenotype, and mutant oligodendrocytes lead to fewer myelinated axons associated with the full clinical phenotype. So that means that having myelin is very important, and it doesn't matter whether it's vacuolated. All right? So vacuolization is not necessarily associated, with clinical signs and symptoms, and is potentially reversible. I would like to give an example of another disease, MLC. You can see there are zillion vacuoles. You can also see that with EM. And I'll show the clinical picture. This is a girl, 4 years old. This is her MRI. It looks horrendous, swollen and diffusely abnormal white matter, and the child is normal. She's a little Dutch guy, a girl, she is normal. This is another patient. She is much older, 21 years. The swelling is much less, and she is wheelchair dependent. That means that there's a disconnect between the severity of the vacuolization and the clinical phenotype. Then we have another form of MLC, which we call remitting MLC, and you can see that it starts the same, and that several years later the MRI is normal, but after normalization of the MRI there are remaining cognitive and behavioral issues and autism, meaning that the normalization of the MRI is not associated with normalization of the clinical picture. So there's a disconnect. I would like to convey that between vacuoles in myelin and the clinical picture. And that means that I didn't even touch it. Do you have another one? All right. That comes with implications for MRI interpretation. And you might wonder why I want to touch a subject, but on with the trials, there are many communications on Facebook and other social media about MRI changes and implications of it. And I would like to point out the limitation of that. So a myelin vacuolization is associated with a high water content and some increase in volume reflected in T2 hyperintensity and aspect of swelling, as you can also see in MLC. Oligodendrocyte pathology leads to less but not absent myelin, and this may lead to some increase in T2 signal, but it is completely overshadowed by effect number 1. And that means that that has implications for assessment of improvement on the MRI. Improved astrocyte function would lead to disappearance of myelin vacuolization, with normalization of white matter water content and loss of volume, reflected in lower T2 signal, so more normal T2 signal and loss of volume. And this change is expected to happen fast. So I think that after starting treatment, you may see soon changes. And I would like you to note that less myelin vacuolization and volume loss are not necessarily good or bad. Improved oligodendrocyte function would lead to more myelin, but that's really difficult to see on MRI, being overwhelmed by this vascularization effect. And this change is expecting to happen more slowly and to be associated with clinical improvement, but poorly visible on MRI. So, MRI interpretation, sorry for the typos, is very difficult in vanishing white matter, and MRI reading should therefore be descriptive rather than interpretative in terms of improvement or decline. And the more relevant MRI method may be the myelin water fraction as representative of compact or uncompact myelin. So we need to work on that, and we will do that together. Okay, my final update is on the Guanabenz trial. That's going to be very brief. As you know, we started the Guanabenz trial in Amsterdam 4 years ago, and we have included 33 patients. 31 completed the trial. We had 2 early dropouts because of the constraints of the trial. So it's participating in a trial is work, and some families, don't find that easy to follow, so they dropped out. And the trial ended May 31, 2025, and the data lock will be soon, and so we will then start analysis. And we are extending the trial 2 to 4 years to allow patients to remain in the trial and get access to the drug. And if effective, we plan to make Guanabenz available and affordable for vanishing white matter patients. So we go for the lowest price possible. This is the trial team. I would like to acknowledge them. Many hardworking doctors on the trial. And then finally, my wishes for the future. I would like to provide general access to the International Vanishing White Matter Registry with governance of the Vanishing White Matter Consortium and Vanishing White Matter patient advocates on who gets to see what and which data are provided. I would love, we have made a core protocol. I would love for all trials to follow the core protocol, most of all because of sharing of data. So sharing of placebo controls means that fewer patients need to be in the placebo group and more patients get access to the drug. Sharing of trial data is something that might be beneficial for patients and families. And in the further future, I would like a trial with 3 arms, Guanabenz, Vosicotivator, and the combination of both to see what that does. And I think that's what I had to say. Thank you so much.
For their help, and also the Saxby Foundation for some of this work. So this is a picture of our lab group. You can see how we're very close with each other, except for like the person in the center there. Everyone's kind of like like a zone of keeping away from me for some reason. Stress granules are a non-membranous organelle. So if you think about there's a cell, and inside the cell there's things like peroxisomes or lysosomes. Stress granules form under conditions of stress, but they don't have their own membrane. They typically form, like I mentioned, when there's stress or when translation initiation is limiting, and they're kind of globs of RNA and protein. They're seen in other neurodegenerative conditions such as ALS, Lou Gehrig's disease, or frontotemporal dementia, and their kind of role and pathophysiology mechanisms is unclear. And so this is just a picture of a cell labeled with a marker for stress granules, and under conditions of increased stress, the components kind of come together and form these little globs. So in various neurodegenerative conditions, ISR activation is a known component of pathophysiology, so we all are focused on vanishing white matter disease, and we have really seen our understanding over the past 10 years of the role of ISR in vanishing white matter disease, but it's also seen in other neurodegenerative conditions such as ALS or Alzheimer's disease, and by modulating the ISR, there seems to be effects on these diseases. ISR activation is also known to lead to formation of stress granules, and so besides just that kind of mechanisms being known, various components of diseases such as 2 different proteins called TDP43 and FUS or FUS are genes that are seen that are mutated in ALS, but also known components of stress granules. So there's kind of this weird linkage between stress granules and neurodegenerative conditions. And so we've been interested for several years now trying to see if there's a role for stress granules in VWM pathophysiology. And so in VWM, there's, as we've kind of discussed, that there's this effect on normal translation, and that there's accumulation of mRNA complexes that then form together into these agglomerations of stress granules. So we wanted to see, is there an increase in stress granules in vanishing white matter disease, and where are they, what's their role in the disease? And so the first kind of experiments we looked at was for a protein called Stalfin. Stalfin is a known component of stress granules. It's known to be kind of a regulator of stress granule formation. And so if you increase Stalfin levels, you can lead to kind of seeding or formation of stress granules, or if you activate the ISR, Stalfin is recruited and helps form stress granules. So the first set of experiments we did was in the zebrafish model that we have, vanishing white matter disease. So in the zebrafish model, which is called ZC102, that's just the name for the mutation in zebrafish, we saw an increase in Stalfin RNA levels. We saw also an increase in protein levels. And then we did some manipulations in zebrafish. First, we did knockouts of the of Stalfin, and we could drive down by knocking out the Stalfin gene in zebrafish. We drove down levels of ISR activation measured by CHOP2, and so under controlled conditions, there's high levels of ISR activation in the zebrafish. If you knock down Stalfin, then you reduce the CHOP2 expression shown right here. Conversely, if you add in Stalfin, so just direct Stalfin directly into the zebrafish, you could drive higher levels, both in wild-type fish and in mutant fish, of ISR activation. So there seemed to be this, so we could see a role for Stalfin in the ISR in zebrafish. And so their next experiment was actually then to go look at what was happening with stress granules. So we looked in the mouse model now, and we saw in the CNS, and so these are, this is just one set of images looking at wild-type mice and the R191H mutant mice. We saw an increase of stress granules in the mutants, which are shown here with the red bars. So we looked in different parts of the brain, such as the corpus callosum. We looked in the cerebral cortex. And so these are pictures showing the increase in astrocytes by double labeling with GFAP, which is a marker for astrocytes. We also did the same thing for oligodendrocytes, and we did the same thing for neurons, of which I haven't shown all the different pictures, but we saw increases of stress granules in astrocytes and oligodendrocytes, but not neurons. The other key thing is that if we made double mutant mice that had both the vanishing white matter disease mutation as well as the mutation for Stalfin, we could then drive the stress granule levels back down. So we also looked at the motor phenotype that we saw the pictures of and the videos of, and by knocking down Stalfin, we could temporarily rescue the motor phenotype. So this is age 6 months, and so wild-type mice are shown in here in blue, and age 6 months is when the VWM mouse mutants first show the motor phenotype, and at that age, the double mutant with Stalfin still had a normal gait, but then over time, the double mutants lost that effect, and so that was no longer protective after by age 7 months into 8 months. So so in addition to looking at the brain, we also looked at other parts of the nervous system, and we saw particularly high levels of stress granules in the cervical spinal cord. So you can see kind of this is just a immunohistochemistry of a wild-type cervical spinal cord and a VWM cervical spinal cord with labeling shown with these kind of red dots. And so in the brain, for example, in the corpus callosum, there was, as I mentioned, an increase in stress granules shown here. In the gray matter of the spinal cord, the mutants actually had a decrease in the number of stress granules, but then the white matter of the spinal cord had this, very, very large increase, much more, even higher levels than what we were seeing in the brain. So we then kind of went through to try and figure out which cell types the stress granules were in. We looked at multiple different cell types, and the most significant increase of where the stress granules were was in a cell type known as microglia. So in mutants there was a slight increase, for example, in astrocytes, but really it was the microglia shown here with the quantification that had the huge increase in stress granules. So, so if we back, so then that kind of, then it's, we're like, oh, wait, what's going on with the microglia? So then the next thing we did was like, okay, what's happening with microglia in vanishing matter disease? And so we saw, so when we went back to look at microglia in the mutant mice, we saw a large increase in microglia in the CNS of mutants. So shown here kind of in the red outline is the corpus callosum, the large white matter tract connecting the two hemispheres of the brain, and the green labeling shows activated microglia, and so this is the quantification of the increase in activated microglia in the corpus callosum. And again, same thing, the cervical spinal cord just had this massive increase in activated microglia in the mutants. So, and so we did multiple experiments to look at activation of microglia. There's different markers. So we looked at different immunohistochemical markers of activation. So this is, again, in the corpus callosum with labeling. It's really this bright green labeling in the mutants that indicates that these are activated microglia. And then we also looked at kind of activation over time. And so at age 2 months, the activated microglia in wild-type mutants are basically the same, but then by age 6 months you start to see this difference in the amount of activated microglia, which then becomes stays high as the mice get older. As another way to kind of look at that, there's different markers that are known, different types of RNA or transcripts that are expressed in activated microglia, whereas there's also ones that are known. And so we saw, for example, like increases in CD68 or ITGAX that are no marker they're markers that show that they're activated microglia, whereas what are called quiescent microglia or microglia that are just sleeping weren't increased, and so it showed again that the mutants had this kind of large increase in VWM. So. So what we've kind of gone through and looked at is we see this large increase in stress granules in vanishing matter disease. We see this, this increase in activated microglia in vanishing matter disease, and the stress granules seem to be predominantly located in the microglia. So we've kind of answered half the question we set out to. So we see this big increase in stress granules. We see them in the microglia, but at this point we're not sure what's happening in the microglia with the stress granules that's contributing to pathophysiology. We'd really like to know, can we normalize it, with treatments? There's the 2B Act that Calico developed, or gene therapy to see if that affects the numbers of stress granules. And then I think with this large increase in microglia, I think it opens this kind of question mark for us whether we need to be looking at the role of microglia in VWM pathophysiology. It was kind of an unexpected pathway that got us to this set of experiments. So, yeah. Thanks for letting me talk.
I have a question. I was reading on the trial page some time ago that there was an expanded use part of that as well. I haven't gone back on the trial page in some time. I had some other personal family things, but I was just curious if that was still available, or is that available, or what's the thought process with that?
You're talking about the expanded access or compassionate use. Yeah, we did. AbbVie opened up and Calico opened up an expanded access program. I think this was initially for one participant who couldn't get into the trial because they didn't meet the requirements. So we did have an opportunity to provide expanded access for that person. I guess that officially opened up an expanded access program because of that. And so I think we have one other participant that is just recently enrolled. But to qualify for that, it's a bit complicated. There has to be a submission from the investigator who is interested in participating and submit an application for review. And for sure they cannot be eligible for one of the trials that's ongoing. So that's the way that works.
Okay, Welcome back. Thank you first to ULF and to the Vanishing White Matter Family Foundation for giving me the opportunity to speak today. I'm really excited to be here. This is my first time at ULF. Will not be my last time though. I've really enjoyed it so far. The opportunity to interact with patients and families has been amazing, and it really helps remind me part of why I do what I do. So today I'm going to talk to you about 3D brain organoids from human patients with vanishing white matter disease. And I think this is an example of a really exciting collaboration between laboratory science, clinical scientists, and then patients, all sort of pulling together to hopefully drive us closer to a therapeutic for vanishing white matter disease. I have nothing to disclose. So because it's my first time here, I thought I would start with just a little bit of an introduction about myself and the group I work with. So I'm a new assistant professor at Case Western Reserve University in Cleveland. I was hired as part of the launch of this Institute for Glial Sciences. This is an institute that is really laser focused on understanding how glia break down in diseases and what we can do to fix those glia that are breaking down. The Institute is led by our fearless leader, Paul Tizar, who I'm sure many of you know. He can't be here today, so I'm sort of here in his stead to present some of this work. And a lot of what I'm going to present today was done when I was a postdoc in Paul's lab. And so, I think we're building something really exciting in Cleveland with this group. And if you want to learn more, we have a website here, and I encourage you to come check us out. And yes, as I mentioned, we're really, our mission is to uncover glial-targeted medicines for diseases. So what does my lab do? My lab is focused on two specific glial cells, of course, being the cells in your brain and spinal cord that are not neurons. There are two glial cells that I'm particularly interested in, astrocytes and oligodendrocytes, which you've heard quite a bit about already. These are the two cells that are central to the pathology of vanishing white matter disease. And like the Institute writ large, my lab is also interested in how do these glia break, and in breaking, how do they cause diseases like leukodystrophies or neurodegenerative diseases. And then if we can understand how they break, can we then figure out ways to fix them? And again, this is going to be a repeated theme, but all of this is with the ultimate goal of identifying glial-targeted medicines. That's really why we do what we're doing. Okay, so how do we study these cells and figure out what's going wrong with them in diseases? My lab likes to use cutting-edge techniques that I sort of put into three large buckets. The first being models. So we use sort of traditional mouse models that many of you are familiar with and we saw about earlier. But we also use advanced cellular platforms from human patients. Once we have those models, we then use sort of big data approaches to try to understand the mechanism of how they are leading to disease. And then ultimately, we want to use these models in tandem with drug screens in order to identify potential therapeutics for fixing the problems that they have. And so I'm not going to talk at all today about the mechanisms or the medicines part of it. What I really am going to focus on is the modeling side and specifically modeling vanishing white matter disease with patient-derived 3D brain organoids. Okay, so what is a 3D human brain organoid? So these are self-organizing 3D brain tissues that grow in a dish. They're derived from human stem cells, and we can take them from healthy control patients. We can take them from patients with a leukodystrophy. Here's a picture of them on the right. They're macroscopic, so you don't need a microscope to see them. You can just see them by eye. These are my fingers on the side to sort of give you scale. And we can grow hundreds of these at a time. I think really critical, these organoids have many of the cell types that are relevant to disease. So they have neurons, which are here in purple. They have astrocytes in red here, and they have oligodendrocytes as well in green. And previously, most organoids that were grown really only had neurons and astrocytes, which is tough for studying leukodystrophies. But Paul's group back in 2018 published the first methods paper for generating organoids with oligodendrocytes. And so I think that's really opened up a new avenue to study diseases. We can't get in and study brains in humans. They're just not accessible. And so now that we have these 3D human brain organoids in a dish, we have a newly unprecedented access to them. And so because of all of these reasons, we really think that these organoids are ideal for studying leukodystrophies. And so we decided we wanted to leverage this technology to look at vanishing white matter disease. To do that, we needed to get access to patient tissue. And so we collaborated with Dr. Ali Fatemi. He allowed us to get access to a number of patients. The one I'm going to focus on most here is a patient with the most common EIF2B5 R113H mutation. And then we also got access to a familial carrier to use as a control for a comparison. So what we do is we got blood from these patients. Then in the lab, we're able to reprogram cells in the blood to stem cells. And then we make those stem cells into these 3D brain organoids. And there's a lot of detail that goes into that I'm not going to go over. This whole process from start to finish can take maybe 5 to 6 months. And so we have our 3D brain organoids. We have one control. We have one vanishing white matter patient organoid. What questions did we first want to ask with these tools? Oh, and thank you. I just want to take a second here to thank patients, caregivers, family members, because truly without you guys, this work would not be possible at all. So thank you. All right, so there were sort of two main questions we wanted to ask first. The first question is, we've made these vanishing white matter organoids. Do they actually mimic key features of the human disease? Because if they don't, they're not a very good model. And so we wanted to look at two things. We wanted to look at whether there's loss of white matter forming oligodendrocytes, and we wanted to look and see if there's increased cellular stress, these sort of two hallmarks of the disease. And so here's an image of the carrier control organoid. We have a low mag image on top and a high mag image on the bottom. Oligodendrocytes are stained in red here, so anytime you see these little green circles with red around them, that's an oligodendrocyte in these organoids. And for the carrier, maybe about 12 to 13% of the cells in the organoid are oligodendrocytes. But when we looked at the patient-derived organoids, I think you can tell from the images pretty strikingly, there were many fewer oligodendrocytes present, and that's just quantified here on the right, a really significant decrease in the percentage of cells in these organoids that are oligodendrocytes. So yes, our first answer or question was sort of answered. We do see decreased oligodendrocytes in the vanishing white matter patient organoids. Yeah. So the progenitors or the mature?
The progenitors mainly.
Yeah, so I should have been more specific. So these are the mature oligodendrocytes stained here in red. These are not the OPCs that in the patients are increased. So yeah, sorry, I should have been more specific. Okay, so the second thing we wanted to look at was cellular stress levels. So I'll just walk you through this graph. Each one of these little dots is a gene that is strongly associated with cellular stress. If the dots are above this line, that means that they are increased. If they're below this line, that means they are decreased. And so you can pretty clearly see that all of these genes are increased in the organoids from vanishing white matter patients. And then we wanted to look a little bit closely at one specific marker, CHOP. So we stained the organoids for that marker. The carrier had very few CHOP-positive cells, where the vanishing white matter patient had a lot of CHOP-positive cells. And again, that's just quantified here on the right. So yes, is the answer to the first two questions. It seems that these vanishing white matter organoids do mimic key aspects of the human disease, and so they are a good model for understanding what's going on. So then what do we want to do next? Because we are therapeutic focused and we're interested in identifying and developing medicines, we then wanted to look at whether current drugs that are being clinically developed for vanishing white matter could rescue these deficits that I just showed you. And so we looked at 2 drugs specifically, one being ISRIB, which you just heard a lot about. This, of course, is the sort of the laboratory compound that we use that has been developed and is now in the clinic. And then we also looked at pridopidine. This is a different class of drug. It has a different mechanism of action. There's been some evidence in the literature from mouse models and cellular models that it could be effective in vanishing white matter disease, so we wanted to test it in these organoids to see if that's true. The first one we looked at was to see if either of these drugs could decrease the cellular stress that I showed you was present in the vanishing white matter organoids. So again, same sort of setup for this graph as I walked you through last time. Each one of these dots, again, is a gene associated with cellular stress, and with them, most of them being down here in the blue below that dotted line, that means that each of the drugs decreased the level of these cellular stress associated genes, with pridopidine being in pink and ISRIB in purple. We looked again at CHOP like I showed you previously, and the staining is pretty clear, right? Here's a vanishing white matter organoid that was treated with just a vehicle, lots of CHOP positive cells. Here's one of the, here's the patient organoid treated with pridopidine. We have significantly fewer CHOP positive cells, and the same for ISRIB. And that's just quantified down here. So both of these drugs could decrease the cellular stress that's present in vanishing white matter disease organoids. And then our next question was, okay, well, I showed you before that there are fewer oligodendrocytes in the vanishing white matter organoids. Here this is again, right, with the carrier here on the left, lots of these mature oligodendrocytes in red. Here's the patient organoid with very few mature oligodendrocytes in red, quantified on the right. What about the 2 drugs? Do they have any effect? And I wouldn't be setting this up if the answer was no, because the answer is yes. So treatment with both pridopidine and ISRIB significantly increased the number of mature oligodendrocytes in these vanishing white matter organoids. And again, that's just quantified over here with pridopidine in pink and ISRIB in purple. So again, the answer is yes to our 2 questions. Both ISRIB and pridopidine can rescue some of the key features of vanishing white matter we're able to model in these organoids. And so this is pretty cool, right? Pretty exciting. We can make these organoids from any patient in the lab. We can play around with them. I can nerd out about the results as a scientist. But why care? You know, if we can't translate this kind of stuff to the clinic, does it really matter? And so we were really excited when Prolinia, along with Dr. Florian Eichler, used some of our organoid data to actually apply successfully for an expanded access study to test pridopidine in 2 vanishing white matter patients. And I'm not a clinician, and this is their work, so I'm just going to sort of give you the highlights of it. First thing is that they saw that pridopidine was well tolerated. I should have mentioned this earlier. Pridopidine has been around for a long time, and it has extensive safety data. This is part of the reason why there was excitement to try it in the clinic. Up to 72 weeks where the patients were on pridopidine, the disease symptoms were either stable or slightly improved. Looking at MRI images over that time period, there was no evidence of significant lesion progression. And then looking at biomarkers, specifically neurofilament light chain, which is a key indicator of inflammation and brain damage, there was either no increase or a slight decrease over the 72 weeks that the patients were on pridopidine. So as a whole, some excitement with this drug. I should mention it's still early days. There's still a lot of work to be done, but I think reason for excitement here. What are we going to do next with these? So what I showed you was vanishing white matter organoids that carry just a single mutation. As we know, there are lots of different mutations that cause vanishing white matter disease. We don't know if they all necessarily would behave the same. We don't know if they would respond the same to the drugs. So one of the things that we want to do is we want to start expanding our panel of vanishing white matter both stem cells and 3D brain organoids. Again, working with Dr. Fatemi, we've gotten access to 2 additional patients and familial controls. 1 patient with a compound Het mutation in EIF2B5 and 1 patient with a compound Het mutation in EIF2B3. And we're still working, still early with these, but we're still working on them. What I can say, and it's actually pretty interesting, right, is that the EIF2B5 compound Het recapitulates what we saw with the R113H compound Het, the data that I already showed you. We see a significant decrease in the amount of mature oligodendrocytes that are in the patient organoids. But that's not true for the EIF2B3 compound Het. There actually was no difference. If anything, there might be a slight trend towards an increase in the number of mature oligodendrocytes in these patients. And so this, I think, sort of highlights how we can start to look at sort of genotype-phenotype correlations using this human tissue that we necessarily couldn't do before. Okay, so just to end with some key points, reminder that we've developed a 3D human organoid model of vanishing white matter disease, and we think this really can help us understand disease mechanisms and then also test therapeutics. These organoids mimic key features of the disease, like I showed you, fewer oligodendrocytes, mature oligodendrocytes, and increased stress activation. And then treatment with certain drugs, the ones that we've looked at so far, are able to rescue those features of disease in these vanishing white matter organoids. Okay, and with that, I would just like to say thank you. I already thanked the clinicians that were involved with the work. I just want to thank members of my lab and Paul's lab that helped do this work, funding, and then also a lot of this was funded through philanthropy, which has been tremendous. And again, thank you to the patients' families and caregivers. Thank you.
My second question is actually related to the model. So how robust are these organoid differentiations, and how much variation do you see between organoids from, for example, the same patient, and how does that relate between the variation you might see between different genotypes?
Mm-hmm. Yeah, so there is...
Please repeat the question, David.
Oh, yeah. Is it on? Okay.
Just for the people that don't have a mic or something.
Okay, so the question was regarding the reproducibility of the organoids and how much variability there is between organoids from the same patient or even across different patients. So there is some variability in the organoids. The differentiation is robust. We always get the same cell types. Usually they're about the same percentage. But sometimes we see differences in the level of stress. Sometimes there's a lot. Sometimes there's not quite as much. There's some variability there. I think vanishing white matter presents an interesting challenge for this because it is a disease that is sensitive to stress. And so any changes in the way that the organoids are grown could potentially activate that. But that also provides opportunities then to understand if the stress is activated, what can we do to fix it. But yeah, there's variability, but they're pretty robust. So.
One quick question, if you don't mind. Very nice talk, yeah. Can you hear me? Yeah. Okay, so following Priska's question, so the cells that you show that are CHOP level increased, what type of cells were those?
Yeah, so the question is what cells in the organoids have CHOP positivity? Yeah. And the answer is astrocytes and oligodendrocytes from what we've seen so far. But we do need to do a little bit more work to try to localize that signal to see if it's in neurons as well, for example.
And the quick question about the drug treatment, how long did you have, so when did you start the treatment? Was that after the organoids were established? And then how long did you have to treat in order to see those changes?
Yeah, so the question is about the duration of drug treatment and when we started. So I didn't have time necessarily to go into this, but you do a stepwise differentiation of these organoids, and towards the end of it is when we add growth factors that push the formation of oligodendrocytes. And it's during that time, it's about a 10 to 15 day window when we add the drugs. So while the oligodendrocytes are being formed, that's when we add the drugs.
For how long?
About 10 to 15 days. Yep.
Thank you.
Mm-hmm. Thank you.
Awesome. Yes, you can hear me. Okay. Hi, everyone. My name is Jessica Herstine, and I am a PhD candidate in Dr. Allison Bradbury's lab at Nationwide Children's Hospital. And today I'm excited to share with you our advancements in gene therapy for vanishing white matter disease. So these are my disclosures. So I know we all know a lot about the basis behind vanishing white matter disease, and Bill filled us in a little bit, but just to kind of recap, this is a loss of function in the giant protein complex called eukaryotic initiation factor 2B, and mutations can occur in any of the 5 subunits, but most commonly it is the 5th subunit, or EIF2B5, that is mutated, closely followed by EIF2B2. So the first patient population that we aim to treat with our gene therapy is EIF2B5 mutations, but we hope to transition this to the other subunits later on as well. So this is what we call our roadmap from proof of concept to clinic. So this is how we follow to get our drug into patients. So first, you really need to understand the disease mechanism, and usually that requires patient samples, understanding on a cellular level what is happening. Then we also need accurate animal models, understanding what is happening in the animals on a multi-organ system, and understanding how all of that interplays with one another. And these two together work for us to understand how this disease is functioning. And then once you understand how this disease works, then you can move on to your therapeutic development, whether that be a small molecule or a gene therapy like we are trying to execute. Once you have your therapy developed, you can then move on to your efficacy and dosing studies. Usually this is in mouse models, understanding what is happening if your gene therapy is working, and then understanding what is your most efficacious dose that leads to the least amount of toxicity. Understanding that in a smaller model helps us translate that to larger animal models. So this is what really helps us allow us to scale it up into patients. So this is your safety and tolerability studies, often needed to be done in non-human primates or even pigs, etc. And once you understand if it's safe and tolerable on that level, then we can actually get it into clinic. So we are all very much aware that vanishing white matter disease is a disease of astrocytes, that they're the most affected. I know we have some other data coming out with oligodendrocytes, which is quite interesting. But from the beginning, we had initiated our study trying to get our gene therapy into astrocytes. So how does astrocytes play a role in myelination? Well, in a normal healthy astrocyte, it will signal, it'll talk to these oligodendrocyte precursor cells, which are like these junior cells, that don't produce myelin, and it'll tell these OPCs to differentiate and turn into myelin-producing oligodendrocytes, allowing for normal myelination. In VWM, astrocytes are sick. They are not functioning correctly. So the signaling process to oligodendrocyte precursor cells is inhibited or prevented. So these OPCs are premature and they're not producing myelin, thereby leading to the loss of myelination. So overall, our goal is to target astrocytes and make them not sick anymore and make them happy and healthy and allow them to function to thereby allow these OPCs to differentiate and turn into myelin-producing oligodendrocytes, thereby rescuing myelination. So we are using a vector called AAV, or adeno-associated virus. It's a virus, it sounds scary, but it is non-pathogenic, meaning it does not cause disease. It rarely integrates, so what happens is it delivers its cargo, which is DNA, into your cell of interest, and it kind of just sits outside of the host genome. So this is very safe, therefore you're not having extra mutagenesis by potentially going into the host genome and causing problems. So it has a really good safety profile. It also has low immunogenicity, meaning that it really doesn't cause much of an immune response in the body. And it has broad tropism, meaning that you can target multiple different cell types, different organ systems, etc., based on the version of AAV that you're using. So when we go to use this in gene therapy, what we do is we take this is just an untouched, normally occurring AAV virus genome, and we take anything that's related to the virus out of it. And we put in key components that are important for driving certain gene expression. One of those being a promoter, which I'll touch on a little bit later, as well as your gene of interest. So for us, we're doing gene replacement, which I'll talk about here in a second, but we're adding an EIF2B5. And all of this is really, really small compared to like the normal DNA that's found in your body. This is like 0.00015%. So it's 4,500 base pairs that we can fit into this vector. So it's really, really small. But luckily with VWM, we're able to do this. You're able to target certain cell types based on your vector design, which I'll talk about with our promoters here in a second. And then certain serotypes or variations of this virus can cross from the blood and into the brain. So this is really dependent on the outer shell called the capsid, which I'll get into here in a little bit. It's easy to manufacture, so this is kind of a benefit of using AAV for a therapeutic. And there's over 300 clinical trials, probably a little bit more since I grabbed this number, that have used AAV successfully. And in fact, there is an FDA-approved AAV therapy for a neurologic disease called spinal muscular atrophy. And in fact, over 2,000 patients have been treated. And this is a version of AAV called AAV serotype 9 or AAV9. And so because that this has been FDA approved and has a high safety profile and a lot of data supporting its safe use, we are using this version of AAV9 to help get it to the clinic faster. So overall, we're using this AAV, adeno-associated virus, to deliver EIF2B5 for treatment for vanishing white matter disease. So as I had mentioned, you can have different versions of AAV, and this is all dependent on the outer shell. So on the inside, you have the DNA. On the outside, you have the outer shell. And the outer shell helps direct it into certain cell types and certain organ systems. And many groups have gone in and tried to characterize or have successfully characterized which organs they are actually targeting. And so what we're most interested in is targeting the CNS, as that's the major affected organ system for vanishing white matter disease. And in fact, AAV9 targets the CNS really, really well. You can also deliver AAV through a number of different ways. The first way is systemic, so just an IV injection of your virus. And the nice thing about this is it's minimally invasive. You don't need to go under any anesthesia. However, you need higher viral doses in order for that virus to go from the brain or from the blood into the brain. And that's because your liver is constantly filtering things out. So you need a really high viral dose in order to get really good virus into the brain. Another way you can do it is a direct organ injection, and many people have shown efficacy with muscle injection, eye injection, even direct brain injection. And a lot of the brain injections are very targeted to a certain region that might be most affected. But this is very invasive, but you don't need as much of virus. Kind of the happy Goldilocks spot is doing a intracerebrospinal fluid injection. So just like a spinal tap, we would go into that cavity between the spinal cord and the spinal column and inject your virus. And this helps direct it throughout the brain, but helps lower the amount of virus you need, making it more safe and tolerable. So I kind of alluded to this, but you can engineer your AAVs to be very specific. And so the way to do that is using specific promoters. What is a promoter? Well, it's like a light switch. So you have a breaker box in your house that can turn on and off like the power to your whole house. We have promoters that can drive expression into all of the different cell types in the brain. And that's what we call ubiquitous expression. It goes everywhere. We also have light switches in individual rooms that can turn on and off lights in specific rooms. And so we have promoters that can turn on and off genes in specific cell types. And so people have actually characterized a number of different promoters to see where it's putting the genes and where it's actually expressing. And so with CBA, that's one type of promoter that expresses in neurons, and we're even able to target oligodendrocytes and astrocytes. And what people have shown is that using these specific promoters, you're actually having high specificity and great expression in the cell types that you aim to target. So we're turning on and off those light switches in specific cell types. So, because we're using AAV9, we know that it goes into neurons and astrocytes. We wanted to make sure that we were targeting astrocytes since they are most affected. So we're using this promoter targeting strategy. We want to turn on these genes in astrocytes. We do have a master switch, which is our CAG. It's supposed to express everywhere. And then we have two astrocyte specific, one large promoter that is GFAP, It's full length, and then one truncated version that's been published and is already in other versions of preclinical gene therapy is called GFA-ABC1D. And to make sure that we were getting it into astrocytes, we did a reporter study. So we put all 3 of these promoters with green fluorescent protein. So anywhere it's expressing, it'll be green. Packaged it into AAV9, since we're using that in our therapeutic vector, and assessed where it was going after delivering it into the spinal fluid in wild-type mice. And what we see here, from that master switch, it really didn't express all that well in the brain, but we had to make this promoter very, very short to fit in the small, small size of our virus. So that's probably why we have less expression in the brain as we would have expected. That full-length, long GFAP promoter targets the neural axis with really great potency, and then that truncated version of that, this guy, doesn't target as well. And so with this difference in mind, and knowing that we are getting significant differences in the green fluorescence over the total brain area, and knowing that AAV is small, and that when you put GFAP with the large EIF2B5 gene, we had low viral concentrations. We still made it, but it didn't make great viral concentrations. So we kind of went back to the drawing board focusing on this difference and realized we could add in about 700 bases, so about double the size of this tiny promoter. And we did exactly that and made a novel intermediate called GFA-1405. And when we did the same exact study, we put it with GFP and assessed where it was going in the brain. We see really great distribution throughout the neural axis, and we were actually able to achieve greater green fluorescence throughout the brain over this GFAP promoter. So we are very excited to move forward with this in our therapeutic design. Additionally, we wanted to make sure that we were targeting astrocytes and that the re-engineering of this promoter doesn't change the specificity. And so one thing that you can do is you can look at individual proteins that are expressed in certain cell types and see if the green fluorescence is with that protein. So in astrocytes, that marker is called S100 beta. And we found that this novel promoter does maintain its astrocyte specificity as it does with the other 2. So we did achieve astrocyte specificity with greater distribution in the brain. So we're doing something called gene replacement. Now, what exactly is that? So that's when you have a genetic mutation which leads to loss of function in protein, just like it's happening in vanishing white matter disease. With gene replacement, and as I mentioned, we're using AAV, we're able to safely deliver a copy of that DNA which sits outside of the host DNA, and then we'll make a functional copy of that protein, and that functional copy will be able to do whatever that normal protein is supposed to be doing in the cell. But we are not touching the host genome. So if that mutation is still being made into protein, that guy will just kind of sit there and do his own thing. So originally, our original game plan before we did our distribution study was to move forward with our therapeutic efficacy with the 3 promoters and EIF2B5 in 2 mouse models, the R191H, which I believe you are all very familiar with, and then a very severe mouse model called the I98M. And in fact, we were able to initiate this study and publish early efficacy results in a last June edition of Molecular Therapy. And if you guys want to talk about that article or want a copy of that, please reach out. I'll be more than happy to share that with you. But we have since added in GFA-1405 to see if we were able to have more rescue if targeting the neural axis more potently would be better. So in the R191H model at the University of Utah under Josh Bonkowski, we were able to assess if the gene therapy was able to rescue body weight. And in mice, unhealthy mice are typically very small. And so with gene therapy, we see that we're able to have an intermediate rescue in the body weight after gene therapy intervention. Another assay we can do is called the rotor rod. Just like the name, it's a rotating rod that you can put your mouse on, and the mouse will walk and walk until it can no longer walk and then fall off. And this is how we measure mobility. And so at 7 months of age, we see that our astrocyte-specific gene therapy, more specifically that really, really small GFA-ABC1D promoter, has the most rescue and mobility, closely followed by 1405 and then our master switch CAG. In the I98M mice, these guys are really sick, and normally untreated, they'll die around 5 months. And if you're unfamiliar with mice, typically an average normal mouse will live up to 2 years. So untreated mice die at 5 months, and then we even did a blank injection just to make sure the injection itself doesn't cause any issues. We have the same survival rate. That poorly packaged, the low viral concentrations does not really extend life expectancy. And what we're seeing here is every divot is a death in the mouse colony. So the steep drop off will show you where that survival rate is at. 1405, that novel promoter that expresses really, really high in the brain, has a mean probability of survival around 222 days, and then CAG is really close to that, so that master switch that's everywhere at 232 days. And then our most efficacious vector is GFA-ABC1D with a 2-fold extension in life expectancy. So instead of 5 months, we're getting a 10-month average lifespan, which is really, really great, and we're really excited to see that. But in fact, we've had animals live up past 2 years of age, which is very exciting. That's the normal lifespan of a mouse. And then we did the same mobility assay in these animals, and we get very similar trends to our survival, with the astrocyte-specific one having the most rescue and mobility, followed by CAG and GFA 1405. Another thing that we can do that's very clinically relevant is magnetic resonance imaging, and I'm sure you're all very aware of this. One technique that we can do to be more specific is we can look at individual regions and quantify the amount of myelin. And when we do that, we see that through multiple different readouts, we are getting an intermediate rescue and myelination in the corpus callosum, which is an area of the brain that's very myelinated. So it's looking like our gene therapy is having an attenuation of myelin loss in our highly myelinated regions. And then because we did our efficacy study, we're moving on to our dosing study. So we have shown you all high dose. We just wanted to see if we were able to get any therapeutic benefit at all. So that's where we see our most rescue and survival with the GFA-ABC1D vector, moving it from 5 months to 10 months. We lessened that 5-fold, so we divided it by 5, and we do get an extension in life expectancy, but it is significantly different from our high dose, as we would expect. When we look at mobility, we don't really see any significant difference, though, in our high and our low dose. When we do GFA 1405, that novel promoter that expressed really highly in the brain and lessen that 5-fold, we do get an extension in life expectancy, but it did not reach significance from our high dose. Yet with mobility, we are seeing a difference from our high and low dose, as you would expect. And then finally, this master switch that should express everywhere, CAG, we lessen this dose as well, and we get very similar trends with our lower dose not doing as well in survival or doing as well in mobility. So if I were to put us in the timeline for therapeutic development with our AAV-mediated gene replacement therapy, I would say we're right about there. We have ongoing efficacy studies, but we're also doing our dosing studies to really see where is the sweet spot for this. And then hopefully soon we'll be able to initiate a pre-IND meeting with the FDA to talk about the potential for moving forward and what they would need from us to get this to clinic. So overall, adeno-associated virus gene replacement therapy is showing promise for multiple leukodystrophies, including MLD, ALD, Guanabenz, and now Krabbe. And we have seen a significant extension in life expectancy of our animals, more specifically from that GFA-ABC1D. And we do see a rescue in mobility as well. So we're very excited to move this forward. And we've shown it in 2 mouse models. So not only the very severe, but the patient orthologous mutation, the R191H. And so hopefully this will help us translate this to the clinic faster. But again, science is always evolving, so there's ways that you can continue to modify your therapeutic design. So even though we're still pushing for this to move forward, there's other pathways we can kind of try and be a little bit more targeted, whether that be AAV engineering or, as we've seen, oligodendrocytes are also important in the disease development, so targeting oligodendrocytes, or even doing combination studies with drugs that are already in clinic for VWM to see if that's not only efficacious, but to see if it's safe. So if patients do want to potentially be enrolled in both, they would have that option. So with that, I would like to thank my PI, Dr. Allison Bradbury, who sadly could not be here today, our whole lab, who has been awesome, and then the Bonkowski lab at the University of Utah, and then all of our funding sources. Without them, this project would not be possible. So with that, I'll take any questions.
Yes, no, doesn't seem that anybody has any questions.
Yeah.
Oh,
Question.
You fabricate brain cells. Right. You make the brain cells from stem cells or something?
You can. We're using mouse models though, so we're doing it all in mice. Mm-hmm.
I see. I just wondered, like, if people donate, you know, their brain, can you all use any of those type of cells, or?
So we can't necessarily do it for gene therapy, but there are lovely banks where you can collect tissue and understand what's happening on the disease mechanism. And then other cell types can be collected, like blood cells, and they can be made into organoids, which then gene therapy could be tested on, which we have a collaboration with the TZAR group at Case Western where we will be testing our gene therapy in their models as well. Thank you so much for that question.
Hello.
Question online?
Can you please repeat the information about seeing the amounts of myelin in specific regions and how that affects things?
Yeah, so what I was referring to was our MRI data. So we do a specific readout called diffusion tensor imaging, and it's very localized. If I can get back to that, here it is. So we were looking specifically at the corpus callosum since it is highly myelinated, and this is just a really good readout on a preclinical level to understand in life if our mice are having an attenuation of myelin loss, since we are doing the majority of our studies a survival study, so we're taking them down whenever they reach our humane endpoint criteria. So this is more of just kind of a preclinical readout to understand how myelination is looking in our mice.
I was just wondering about the, very nice, thank you, about the stoichiometry of all the subunits.
Yes.
And whether, you know, GFAP is a strong promoter, so maybe it's going to generate a lot of the EF2B5, and, you know, what the cell is doing with all this excess subunit,
Yeah.
and also whether that's going to, you know, as the assembly of the decamer, it's going to be, I don't know if you looked into this,
No.
but like, yeah.
That's a great question. So the question is actually referring to the protein complex. It's huge. There's 10 individual subunits, and how would adding just one of those affect how it builds itself? I am looking at that because I'm very interested. I have a feeling that, I don't know if it will affect the stoichiometry, but Carmilla Sadrowski did a lot of the in vitro work, and I'm going to be doing some transfections and looking at how our promoters are driving expression, and concentration dependent leads to different outcomes. So, great question.
Hello. You said you're going to target EF2B5 first.
Mm-hmm.
What is the time frame to target the rest of the mutations?
That is a great question. So it really comes down to the FDA. Right now, EIF2B5 has all of these models. So we have the platform to test efficacy in vivo, in mouse models. I know EIF2B4 has a mouse model. So it really comes down to if they would allow us to translate it on more of an organoid level, or if we actually need to generate mouse models and test each one of them individually. I think if we could also show safety data on a larger animal model where it's not necessarily affected with VWM, but at least shows safety and tolerability, they might let us move forward. But it really comes down to that conversation with them. Yeah.
I have a question, if that's okay. So what are the mice dying from, and why do you think some survive and some don't? Because you have some that die quite quickly, I mean, even if they're treated.
Yes. Yeah, we have a lot of variability in our survival. From the untreated standpoint, these animals, at least on the I98M side, They lose body weight, and then they lose the ability to ambulate. So that's when we call it their humane endpoint. We do see some seizures in this animal model. It's the only VWM model that has seizures. And so after gene therapy treatment, we are seeing an extension in life expectancy, improvement in body weight, et cetera, but we haven't fully corrected the seizure phenotype. And that's one thing that these animals, they have a seizure that lasts too long, will go into respiratory failure. So we are starting some potential combination therapies. I'm interested in trying some sort of anti-seizure drug, potentially with our animal model to see if that alleviates it. But I am also looking at some other molecular signatures to see if certain cell types are dysregulated and if targeting astrocytes is not the only way to do that. So as of right now, for the therapy side, they've been dying mainly of the seizure phenotype, but I have some things in the works to hopefully correct that. Yeah, so with that, because they're not reaching our humane endpoint, we don't have a lot of tissues to work with. I have 2 animals that I was able to catch right after like a death event, and I'm going to be looking at the pathology of those. I also did single cell sequencing on one of those animals, got good viability, and so I'm interested to look at the molecular signature. I'm really not sure what to expect on the pathology side of things, but it would be very interesting to see.
Great, thank you so much.
Thank you.
Presentation to come up. I'm going to be super quick. I'm Allison from the VWM Families Foundation, in case you guys haven't figured that out. And I just was going to go over kind of our, numbers for our families mostly. I don't think you guys, scientific people, will be very bored by this, but I'll be very fast. Yeah. Well, we have lots of time, so. But also, I don't think anybody really wants to hear me talk. Oh, look, there I am. All right, here we go. Oh, gosh, did I do that or did you do that? All right, so just quickly, we're going to go over the numbers. We've been around for 9 years now, which is kind of amazing. I can't even believe that. So in total, we have raised actually that's a little higher because I do have a check at home that I haven't deposited yet. So we've raised about $650,000 in total. Almost all of that, $489,000, has gone to funding research. We've cost of goods sold, we've had some fundraisers, stuff like that. The operating expenses, this one I wanted to go over with people. That's a large number because that is a retainer for a lawyer. So because we are now working more with pharmaceuticals and we're having contracts that need to be evaluated, and so that's almost all of that is lawyer retainer. So our operating expenses are generally incredibly low. So 30,000 of that is just the lawyer, and 6,000 over 9 years has been our operating expenses, which is almost nothing because we're all volunteers. So and also just FYI, these come out of a separate pot of money. This comes out of a separate pot of money, so your donations are still going directly to fund research. So we have a separate funding stream that helps pay for those operating expenses. So as of now, we have done all the donations. We have $90,000, and we owe you 50,000 euros. I think we have one more payment, and so we will be paying that this year, and then I think our commitments right now are over, so then we have a little bit more flexibility for where to spend it. But this is just a breakdown of where the money has gone over the last 9 years. The vast majority has gone to the Guanabenz trial at this point, and then also we've been doing helping with some of the gene therapy, which we'd love to keep helping funding that, because we feel like that's kind of the next phase in terms of treatments for VWM, is looking at the gene therapy in conjunction with the drug treatments. So this is it's Brett, Donna, myself, Nick, and Erica, who couldn't be here, and Margaret help out with a lot of what's going on the team. And then I just wanted to say a huge thank you to Brett and Donna, who organized all of this. I did literally nothing but showed up this morning. So thank you, because without even knowing what was happening. So they did all of the organizing for this, and they've been doing it for the last few years, and so grateful for everything they do for the foundation. And also, I just wanted to pull out a few people have done like a ton of fundraising this year. So Angel's Family, Dax, Reed, and Nash have all done quite a lot of fundraising, which is we're super grateful. And we need, you know, even though we've kind of reached the end of our contract for Guanabenz, there's still a lot of work to do and a lot of funding is still needed, both in Guanabenz and gene therapy, and any help we can get with fundraising, we would very, very much appreciate. And then the last thing I wanted to say, if anybody, some of us who are running the foundation are getting old, and we really could use some, you know, families with younger kids to get involved, because we'd love to be able to kind of hand over some of the responsibilities to sort of the next generation of families. So if anybody is interested, please get in touch with myself or Brett or Donna. I'm sure all of the families know where to find us, and we'd love to get more of the younger families involved. And I think that's all I have to say.